Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this section and following the biology syllabus, you should be able to describe the factors that affect the growth of bacteria. What does this actually mean? What are we trying to understand? Well, this is very straightforward. All you have to be able to do is to be able to describe the things that affect how bacteria grow. Let's look at bacterial growth factors. Before we study the factors that affect bacterial growth in any great detail, there are a couple of points that we have to understand. First of all, when we're talking about bacterial growth, what we actually mean is an increase in the numbers of bacteria, not an actual increase in their size. We also need to realise that bacterial growth is going to be affected by their access to food and by 5 environmental factors. Too much or too little of any one of these factors will slow down or stop growth. At the end of the day, the factors can be connected or are related to how bacterial enzymes work. The factors that affect bacterial growth or an increase in the numbers of bacteria are temperature, oxygen concentration, external solute concentration, pH, and pressure. As a memory tip, we know from our work that the germination of seeds are affected by TOM, meaning suitable temperature, oxygen, and moisture. As a memory tip here, the factors that affect bacterial growth are quite similar, but we have an additional 2 factors. The factors that affect bacterial growth are TOM, PP. Suitable temperature, oxygen, moisture, which is related to solute concentration, pH, and pressure. We will now look at each one of these factors in turn. Temperature. Low temperatures slow down the rate of bacterial growth. This is because low temperatures slow enzyme activity. And when enzymes are working slowly, bacteria will grow slowly. This is one of the reasons that we can store food in a fridge, because the enzymes in the food and in the bacteria of decay work slowly due to low temperatures, and therefore the food will be preserved in the fridge for a longer period of time. Most bacteria grow well between 20 degrees Celsius and 30 degrees Celsius. Most bacteria are denatured at high temperatures. This means they lose their shape. Now, of course, there are always exceptions, and thermophiles are bacteria that can tolerate high temperatures and are capable of living in hot springs. To understand how temperature affects bacteria and bacterial enzymes in more detail, it might be a good idea to go back to the enzyme playlist. If you recall, the lower the temperature, the slower enzymes work. The higher the temperature, the faster enzymes work. When the temperature is too high, the enzyme changes shape and stops working. An enzyme with a changed shape is denatured. So you can see the connection between this material that we've already covered in the chapter on enzymes and the growth of bacteria. The second factor that affects the growth of bacteria is oxygen concentration. Most bacteria need oxygen to survive. These are aerobic bacteria. They need the oxygen to produce energy during respiration. The energy is usually produced in the form of a chemical called ATP. This is why oxygen is sometimes bubbled through bioreactors when bacteria are being used to make a product. So for example, if aerobic bacteria are being used to make yogurt in a bioreactor, then oxygen is bubbled through the container. Don't forget, bioreactors are containers in which living cells are used to make a product. A very important definition from our previous work. For those of you able for a little bit more, bacteria can be classified according to their oxygen requirements, because bacteria are very sensitive to oxygen concentration. There are 4 different classifications. The first classification is aerobic bacteria, or aerobes. These bacteria need oxygen for respiration. An example of such bacteria are streptococci bacteria, which cause sore throats. This is logical, really, because they have a ready supply of oxygen when they're living in your throat. The second classification are anaerobes, or anaerobic bacteria. These bacteria do not need oxygen for respiration. An example of such bacteria is Clostridium, which can cause infection of our intestines. Third classification of bacteria based on their oxygen requirements is that some bacteria are facultative anaerobes. This means they can carry out respiration with or without oxygen. In order to help us remember this, if we look at the word facultative, it reminds me of the word facility. So I think to myself that they have the facility, they are able to carry out respiration with or without the oxygen. An example of such a bacteria is E. coli, which is often responsible for causing food poisoning. The fourth classification of bacteria based on their oxygen requirements is that some bacteria are obligate anaerobes. This means they can only carry out respiration without oxygen. In fact, the presence of oxygen inhibits the growth of such bacteria. Again, as a memory tip, I remember this. If we look at the word obligate, it reminds me of the word obliged. So these guys are obliged to carry out respiration without oxygen. Clostridium tetani, which causes tetanus, is an example of such an obligate anaerobe. The third factor that affects the growth of bacteria is the external solute concentration of the environment in which the bacteria are to be found. Now, this is a very important point and is linked into the section on osmosis. If you haven't done osmosis before, it might be a good idea to look up the osmosis videos. Now, we need to be very clear here about our terminology. It is easy to mix up the vocabulary. We're going to stick with the idea of the definition of osmosis and focus on the concentration of water. Now, the concentration of the surrounding solution in which the bacteria are to be found affects bacterial growth, because depending upon the concentration of such a solution, the bacteria will either gain or they will lose water by osmosis. And such a gain or a loss affects enzyme activity and thus affects bacterial growth. Now, we need to understand that a large concentration of solutes, in simple English, a large amount of salt or sugar, is the same thing as a concentrated solution. So a large concentration of solute is a concentrated solution. But for the purposes of the exam and the purposes of the osmosis definition, we're going to look on such a solution which a large amount of salt or sugar, we're going to look on that solution as being the same thing as a solution with low water concentration. Now, low water concentration, not leaving out the word water, is a very important phrase which we use in the definition of osmosis. Equally well, we need to understand that a small concentration of solutes, or a small amount of salt and sugar, or a dilute solution, is the same as having a high water concentration. Not forgetting the word water again for the purposes of the osmosis definition. You could look on undiluted or concentrated my waddy straight out of the bottle as being a concentrated solution or a solution of low water concentration. Doesn't matter the amount, whether you have a glass, a bottle or a bucket of my waddy, if it is concentrated, it is still of low water concentration. You could look on a diluted glass of my waddy as being a solution of high water concentration. And again, if you have diluted your my waddy, it doesn't matter whether you have a glass of it, a jug of it, or a bucket of it, it is still of high water concentration. Now, we need to be very clear on this terminology, which should be revised with the osmosis videos, as I've already said, before we will be able to understand the following concepts. Now, let's remind ourselves of the definition of osmosis. We've already learned that osmosis is the movement of water from a region of high water concentration, meaning low solutes, a small amount of salt and sugar, high water, in other words, a dilute solution. So according to the definition of osmosis, water will move from a region of high water concentration to a region of low water concentration. Low water concentration means a lot of salt and sugar or a high amount of solute. How we remember that is that we come down from a high. The water will move from a region of high water concentration to a region of low water concentration. Or those of you who would prefer the alternate wording is the water will move from a region of low solute concentration to a region of high solute concentration across a semi-permeable membrane. But I would really stress that we learn the definition and we use the vocabulary connected with water concentration. And as we know, of course, osmosis is passive. That means it does not need any energy. Now back to the idea of how solute concentration can affect bacterial growth. If we take the example of bacteria in low solute concentration, meaning, as we're going to stress, high water concentration, then we should know from what we have just revised that water will enter the bacterial cell by osmosis. We come down from a high, moving from a high water concentration, which is outside the cell or external to the cell, to a low water concentration inside the cell. Now, don't forget, of course, that the water is going to enter the cell by osmosis, moving from a high water concentration to a low water concentration inside the cell through or across a semi-permeable membrane. This means that the cell will expand as the water enters the cell and enters the cell cytoplasm. However, the cell wall will actually stop the cell from bursting. Most bacteria live in less concentrated solutions. Now let's look at the scenario where bacteria are to be found in a solution of high solute concentration, or in other words, low water concentration. Now in this case, water will leave the cell by osmosis, moving from a high water concentration inside, high water inside means low solutes, to a lower water concentration found outside the cell across a semi-permeable membrane. The cell will therefore then be dehydrated, and as a result, the enzymes will stop working, and this will have a knock-on effect on the growth of the bacteria and their reproduction, because the bacterial cells will shrivel up and die. Adding salt, which is also known as curing, and adding sugar to food therefore allows it to be preserved, because the bacterial cells are dehydrated, the enzymes stop working, and the bacterial cells stop growing. Recall the video on osmosis, where we already learned how food preservation works. When microorganisms land on the food, like salty meat and jam and marmalade, they lose water by osmosis. The microorganisms shrivel up and die. They cannot decay the food, and so it is preserved. The fourth factor that affects the growth of bacteria is pH. pH affects enzyme activity, and thus pH will affect metabolism, the chemical reactions that go on in bacteria, and thus the pH will affect growth. Recall that the pH scale is a measure of acidity or basicity. The scale ranges from 0 to 14. A value of 7 on the scale means that the substance is neutral. A value of less than 7 on the scale means that a substance is acidic, and a value greater than 7 on the scale means that a substance is basic. So the pH, or the acidity, of the solution in which the bacteria are to be found will affect bacterial growth. Most bacteria grow at or near neutral conditions. Most bacteria cannot grow at a low pH, or they cannot grow in acidic conditions. This is the reason why foods can be preserved by pickling in vinegar, because vinegar creates an acidic environment in which the bacterial cells or the bacterial decay cannot grow. Lastly, pressure affects the rate at which bacteria grow. Bacteria can actually live in a large range of pressures. Some bacteria can live very deep down in the ocean, down in deep sea vents where you have extreme pressures. However, the growth of most bacteria is inhibited by high pressures because the bacterial walls collapse and break. Pressure-tolerant bacteria for use in bioreactors have been formed by genetic engineering. Don't forget, bioreactors are containers in which living cells are used to make a product. So, in a nutshell, the factors affecting bacterial growth or an increase in bacterial numbers are temperature, oxygen, external solute concentration, connected into the idea of moisture or water concentration, really, pH and pressure, to be remembered by the little memory tip, Tom P. P. And there you have it. Practice in a jotter. Factor in the work. Now that we have reached the end of our lesson, have we achieved our objective? Can you describe the factors that affect the growth of bacteria?